So welcome to PMTC Knowledge Day 2022. Lovely to be back to in-person events, and actually this time around we have a hybrid event, so welcome to those who've made the journey here today and also to our virtual audience. So really delighted to bring Knowledge Day to you today. You've got a programme that sets out, what we have in store for the day, and so we're really excited to share our research with you, and particularly here in the Brunel Institute, which is our base here at the University of Limerick. So, as many of you know, the PMTC, as an Enterprise Ireland IDA Technology Centre, is funded really to support that bridging between industry and academia. And we're very much enterprise-led, so we're really trying to support the pharmaceutical sector in Ireland, increase its global competitiveness for Ireland, and we do that through high-impact applied research. You'll see as we go through the day, we've got three main research themes. These are all informed by industry. Indeed, the projects that we perform are very much informed by industry and industry actively participating. And this year we've got some panel discussions after each session where we're bringing a lot of our industry members to share their insights and their thoughts with you. So industry isn't the only thing that's important. I guess, we are bridging the academia industry paradigm, and the academic role in research is really key, and I guess maybe that sounds an obvious thing to say. I think particularly for this sector, and so, thinking about the framework in which we work, this regulatory framework, there's a bit of a power imbalance that's maybe at play in terms of the role of the regulator compared to industry, and for certain topics there can be sensitivities around those that may be perceived barrier towards advancing, particularly with new technologies. And I think the role of academia here is really important in that it can have a balancing type effect. So if you like, the academic doesn't have skin in the game, can maybe push the envelope a little bit. And I think even by its very nature, that's a great support to the industry. So that's what we're looking to do, to deliver this balance, influence where we can, to try and support change that's really advantage to the industry and to the patient at the end of the day, and to provide that independent perspective to the work that we do. So the centre has been around since 2014, and in the early days we had members in single figures. We're now in year 4 of our phase 2, and happily we have really grown in terms of numbers, 38 members today. And a lot of what we're going to talk about in the early part of the day represents the research that we've had ongoing through our phase 2. But as you can see, we're hoping to go into a phase 3, and that's not too far away. So already we have an eye on what is it we're going to do in the future. That future state is to be determined, but we're looking to garner the insights from industry and really have that collaboration in shaping that path forward. So we'll touch on some of those things in the later part of today as well. Not only have our members grown, our affiliated RPOs have grown, and you can see the entire community here, the industry members, both multinationals and Indigenous SMEs, and it's really that richness and that diversity within the membership, I think, that brings a lot of benefits and indeed opportunities to share knowledge and to work together and really try to advance both the multinational sector and the Indigenous SME sector together, and of course supported by the academics. And I guess as a national centre, what we're seeking to do is make sure that we're tied into a number of different RPOs, and that on a given project we're attracting the best academic talent to support and feed into that project to maximise our results. And of course our team. So our team in PMTC has grown as well. In our phase 2 we have anchored many more researchers here. So while we're still a national centre and we have a number of affiliated RPOs, we do have critical mass, I would say, here within the University of Limerick. And you'll see that as we go through indeed from our speakers today, and I'll give you a snapshot of our core research programme in phase 2. So I think this is a great basis for further growth for PMTC as we continue to build our capabilities, both in terms of our people and in terms of our infrastructure. And that brings me on to my next point. So we, based here at the Brunel Institute, this is a world-class research institute and has a lot of capabilities that have been gathered over time, across a number of research groups. And so we have a lot of pharma-centric capability, which I think gives us something of an edge. And we're continuing to grow those capabilities, particularly in the areas of process analytical technology, which is very close to our hearts in PMTC, and looking at both large and small molecule in terms of those capabilities. We're looking towards the future in terms of digital. So a lot of the newer infrastructure that we have acquired is, of course, digitally enabled, and this is to keep in step and make sure that what we're working on is representative of what's in the field. We've grown our team on data analytics, and you'll hear more about that today. And of course, this is one of the foundations, if you like, for the digital journey that all of our members, both SME members and multinationals, are going on. And then particular call out to a significant piece of kit that we acquired through the Enterprise Ireland capital call, an ATEX CIP system, which again is suitable for both large and small molecule research endeavours. You'll hear more about that later on today. So I mentioned earlier on we have three main research areas. Pharmaceutical plant cleaning is one, process optimization and control being another, and data analytics, new but ever growing in this phase and certainly into the future. So I guess we've got a lot of outputs on these. I'm not going to go into them in a lot of detail. Today you'll hear most of what's happening within our phase 2, but some of these themes, particularly cleaning and process control, those have been carried forward since the earlier phase. And so this is what we're trying to do as we move forward as a centre, grow in the capabilities that we have and expand in step and in line with the industry need. There's a lot on here, so I'm not going to speak to it, but just to give you a sense of when we think of our core research, we stand up projects under each of those three themes. A lot of activity happening here in the University of Limerick, but also our collaborators in MTU, Mike McAuliffe, you'll hear from Mike today, equally TUS, our neighbours. John Cosgrove will speak to a new project that's starting, and we have a significant data analytics project. We have our own senior research fellow will speak to that, but we collaborate with Tech Centre IMOR on that one. So we have a lot of core research, which is our state-funded research ongoing, and you'll hear the details from our experts here today. Beyond our core research, we're also growing in terms of other avenues of research that we do, but keeping it within our capabilities and our research pillars. So we have a number of IPPs with a range of companies across each of them, and indeed contract research. And I suppose this is really just to illustrate that there's a lot of ways that we can support industry. And for this type of research, it doesn't require membership. So while we have 38 members, there are many, many more companies that we're doing work with, and different ways to access these capabilities within our research centre. So our highlights then, and I'm really proud to say that we've those 38 members. We have over 200 participating companies, so those are companies doing research with us, attending events, workshops and knowledge sharing, 85 million expected impact, and that's from a measurement done by an independent panel. And you can see there we've had huge amount of growth in terms of our postdoc researchers and PIs and RPOs. And indeed events like today, which are really key to our mandate, which is to try and help grow the sector and support through our knowledge sharing to disseminate the work that we're doing. So over 1,500 attendees, and that's just in our phase 2. So I think, we've had our challenges through COVID, as every organisation has had. We've done a lot through virtual platforms, but there's no substitute really for face-to-face. So, I hope we grow on that, and I hope we attract more and more visitors, both at events and to lab visits here to the PMTC. One more recent and very significant achievement, I'd like to just recognise the team. So we've recently won, through the Pharma Industry Awards this year, Research Centre of the Year, and for that ATEX rig, small project of the year. And I suppose for that one, I think, while the Research Centre project, or the Research Centre of the Year, is recognition of the whole centre, and that's across a wide stakeholder group. The small project of the year, I'd like to give particular call out to former senior research fellow, Rabba Morris, who's now our research centre manager. So Rabba was successful in securing funding for this through the Enterprise Ireland capital call, but then led the project, which was complex, and we had to pivot quite a number of times through that. not least because we had anticipated vendors would do the design. That didn't happen. We were supported internally through one of our team members, Nicholas Aramuni, and also our academic lead, Vivek Ranada, to pivot and to absorb that design and ultimately deliver the project. So and RS Shard. So, you know, I suppose a good example of how we can be agile and pivot and draw on those expertise as and when we need to. So really proud of that achievement and of the team in general. So maybe before I finish and hand over, I'd just like to give a call out as well for the team organising today. I think you'll agree it's a really good, well-organised event, and in particular, Carol Ryan, who has thought of everything, including signage and all those practical things that maybe the rest of us don't think. So really looking forward to a great day ahead and a lot of dynamic discussions. I'd like to encourage the audience to participate. So we have both our virtual and physical audience using the QR code as we go through the day. We will have some polling questions for the members, and as we have our panel discussions, we'd encourage questions from the audience and really try to make this as interactive as possible. You have a booklet which shows the agenda for today. I'm not going to get into it. Hopefully you've got that physically with you if you're here in the room, or on a PDF if you're in the virtual audience. We're probably a little bit behind time. We had a slightly late start. So with that, I'd like to hand over to Raba, our Research Centre Manager, to begin to talk to us about some of the core research in PMTC. Okay, I'll just close
that. Thanks, Sarah. It's really a pleasure to be here and talking to the PMTC Knowledge Day. And as Sarah mentioned earlier, being in face-to-face meeting is really great, and welcome to everybody. Well, I have a few slides that Sarah has already talked about, so I will escape those and save some time maybe in the agenda. Anyway, what I'll be talking about here today is the cleaning theme that I led before in the center before moving to my new role. That's the cleaning theme. We'll have 3 talks, myself, Nicholas, and RS Chard, and we'll tell you how we did the work and talk about the impact that we have achieved through the cleaning theme, basically. So my talk will be around this outline, just talk about the cleaning and the impact we achieved today, the cleaning core project Spotless, and just a few words on the PMTC Good Cleaning Validation Practice Guide. And then I'll just finish with the conclusion. Yeah, that's a few years back. I think we had that survey that shaped our cleaning themes within the center. And the question that was asked, what are the issues are facing the industry, pharma industry in Ireland? And you can see there are 3 themes, which are the cleaning, process, and data, where cleaning came on the top, which is about 44%. So I'll just focus on the cleaning. Well, we talk about how we use fundamental science to help our industrial members to develop more efficient and sustainable cleaning process, and then how we do the process optimization and scale up. So through the years, I think we have achieved really a good great impacts. Well, we reduced the changeover by an average of 2 weeks, and then changeovers about 10% in a year. And we've been able to reduce the usage of solvents about 40%. And all those outcomes, they really have a great contribution in increasing production capacity and competitiveness of the pharma sector, and they will feed straight away in the more sustainable and efficient processes. I'll just go through our core project and tell you how we achieved those impacts. As Sarah mentioned, there's many funding streams and ways how we can collaborate, including the Enterprise Ireland fundings and contract research, as well as using the research components from our members to do the core research. And Spotless was one of the first approved projects in this phase, which is about how is modeling and experiment to design cleaning rigs, so where we can do the optimization and study all the fundamental sides of the cleaning, and then how we introduce and use the PAT tools for real-time monitoring and verification. So cleaning is how our approach in PMTC is how to go from this state to this one in most efficiently, in less time, and will be efficient. So that's in a restaurant or in a kitchen, but in real time, this is what you can get after a process. Basically, you can have those dirty vessels, and it has to be cleaned in a efficient way. Look into the safety issues as well. So our approach in PMTC is built in three pillars. So that to have really a fundamental understanding of the pharma cleaning. So it's based on, first of all, look into the surface science, how the surface of the equipment and different materials interact with the residues, and how those residues are adhering to the surface, and what's the best way to remove them. We look also to the chemistry, either for the residues that we are trying to clean, or even the chemistry of the cleaning agents, if you are using organic solvents or detergents or aqueous solutions. And also we use the help of the CFD modeling to design or to find out the best way how to deliver those cleaning agents, and what are the flow rates, what are the cleaning times, and so on. So we start with in our methodology by the modeling and trying to screen for the best cleaning agents based on solubility. And to do that, we use some computational techniques using COSMOS or Hansen solubility parameters for fast screening. And then we use the CFD modeling to inform us on the optimum delivery methods. After we select our cleaning or the best cleaning agents, we go and do the experimental side, and for that we measure solubilities. Also we characterize our residues, and then we do the cleaning trials and see if our recommendations are good, and we have achieved what we wanted by doing that. And we have the privilege to be in Bernal and at the University of Limerick. We have the access to the best some of the world-class state-of-the-art characterization suites, so we can do the complete physical-chemical characterizations on the residues before and after the cleaning trials. So we would like to have a time zero, basically. We stamp that we would like to know how our residues look like, and then we do all those characterizations, so we know the time zero, basically. We do our selection, our screening for the best cleaning agents based on solubility, and we can have a trend using the temperature. So basically, you may increase your solubility by a factor 2 or 4, moving from 20 to 30, 40, or 60 degrees, if possible, to use that within the cleaning process. We do our measurements as usual. We select our solubilities, and then we check also for the stability, because it's really important to keep the integrity of the residue, because if there is any degradation or any chemical reactions, if we are doing the verification using the offline method like HPLC or others, maybe we'll be looking for different molecules. So we have to be sure there's no degradation of the molecule happening. So one of the objectives of the Spotless project is there a way to have a model that would help us to cluster for example, all the chemicals and the solvents, and then before going to the lab and doing any experimental measurements, can we predict the solubility and cluster all those chemicals in the more efficient, accurate way? And we have done that, and we are really happy with the team because we achieved that during this project. We'll be able to predict the solubility of residues of chemical entities by 90% accuracy. And this is just by looking to the physical-chemical properties of the molecules without doing an experiment, basically. So I can show you here the results with the predicted versus the computed ones, and you can see R square is about 0.91%, and that's really great achievement. In the middle figure here, where you can see that we can classify a good number of molecules according to their solubility using the pharmacopoeia scale, going from slightly insoluble to very soluble. And that was really good. So we did that for over 550 molecules in 21 solvents, but we just selected here 10, the common ones that are used in pharma for cleaning or for process. And then if we take just another example on the molecule, if let's say we would like to use methanol for cleaning, so we can cluster those molecules according to their solubility. So then we have a great idea before going to the lab and selection. This will reduce huge time in optimizing the cleaning methods. And of course, we have already a database of, as said, 700 molecules in 21 solvents, common solvents used, and these are available to our members through the Spotless project. We also built as a test and validation of the database, which is about over 10,000 molecules that has been used in simulation, but in this case, we just used water. But it's possible to do the same thing for organic solvent. And then after that selection, we just go and validate that experimentally. And then we did that on a few molecules from our partners. And then we showed there was enhanced the solubility and the cleanability of those molecules. I'll show later on the cleaning trials via developing mixtures using binary and ternary systems. After we select the cleaning agents that we think is the best, we just do our cleaning trials. And what you can see here as the recommended cleaning using the modeling and the experimental are outperforming methanol. That's the common organic solvents used in cleaning. And you can see we clean the coupon using acetone in 11 seconds. The ethyl acetate acetylnitride is 24, but you can see methanol took that. It's about 440 times, 481 seconds. The other objective of the Spotless was the development of an online technique to monitor and verify the cleaning in real time. So we tested different techniques using UV-Vis, Raman, FTIR, NMR, and then we found that the UV-Vis is the best tool to use because it's the more sensitive, and we had a low LOD 0.1 ppm and LOQ for 0.4 ppm. So we opted to use that, and we designed our cleaning film flow rig, designed using the CFD, thanks to OREST for providing all those data on the scale down to have the right size of coupons and the right flow rates that can be scaled up later on and used in plants and cleaning the big reactors. Of course, that's what you see in the fume hood. That's what we can use in a research lab. But if you are in a plant, you would like to have a better, system. And in a collaboration with one of our members, multinational members, and SME at her present here and technology, we developed a smaller system in a suitcase that can be carried out everywhere in the plant. It can be in the QC lab. It can be also taken on the floor and used for cleaning verification, for rinse recovery as well. Depends on the application that you would like to use. I'll just give you here an example how we use the film flow rig combined with the PAT for real time. While we are cleaning, we are measuring at the same time the concentration of the dissolved molecules. So we are reading in real time the concentration, and then you can see this is the profile. We integrate that, we get the amount of the cleaning. So one observation here that you can see when we finished, we take our coupon just by weight. We can find how much API we removed, and you can see that the PAT recovery was much more or less. And actually, at the beginning, you said that maybe the PAT is not working, but it turns out it's great results, and this helped us to have more information on different cleaning mechanisms if we are it's not just dissolving the molecule. What I've seen is there's a lot of lift-off, and this may happen in the gross clean. So we developed another technique using an optical camera to quantify both, basically, how much we remove by dissolution and how much we remove just by shear-off. And Nicholas will talk more about that because he used that on the optimization of the cleaning parameters. And of course, we double check and double check every time. There was no effect on the molecules. There's no degradation, no coloration, or any polymorphic changes. So we can use, a fast measurement using Raman straight away. It takes few minutes, or we can go to the SEM and get more pretty pictures and see there's nothing has happened. So this is our learning curve in the lab, but we would like to optimize that and take it further to be applied in the plant. So as I said, we use the two techniques. The first one is the CFD modeling, and the team working on that, help us in designing on flow rates, weightability, estimation of the cleaning rates, and by dissolution in film flow, and then of course using the outcome for the scale up. And in analogy to the Sinner circle, what we do, we can also optimize that using the film flow rig, where we can change different parameters. We can, the mechanical, the flow rates, or the chemistry, different concentrations or different combinations of organic solvent, or maybe the temperature. And this allows us basically to find the sweet spot, really, where we can use less time, less detergent or less organic solvent, less heat, and get the best outcome. If you see on the second figure here, we can see that with those parameters, the optimized parameters, we've been able to clean at really less time and low temperature, 20 degrees, less concentration of detergent, and we are below the VRLs that we have measured in the lab. And once we have done that, we have to scale up, and we use the pilot plant, a CIP rig, and Sarah has mentioned that. We're really proud that this project won the Pharma Award. And thanks again for the funding from Enterprise Ireland that allowed us to acquire this unique facility, and that we offer to our members, to access, either to optimize their cleaning methods or to develop new cleaning methods, and using the film flow rig, and then scale it up to the rig, and it's modular and can accommodate different type of cleaning agents and the different materials. Just the last slide. I think I'm on time. So there was a call a few years back to have a guide to help our industrials to summarize all the rules and for the cleaning validation, and we had a team that had worked on that. And I would like to thank them all. So there's an update of that version, the second edition. It's under review with HPRI, and hopefully we'll get it, early next year, and that will be distributed to our members. Yeah, that's my conclusion. Finishing up. So we have developed a database for clustering chemistries and cleaning agents that is accessible by our members. We have combined the CFD and experimental to understand different cleaning mechanisms. We use, by the help of using the PAT technologies, and then we have designed a film flow rig. Actually, we are using it here, but we are happy to give it to our members if they want to use it in their plants and to design their own cleaning methodologies. And of course, lately is the design of the ATEX CAP rig that's also accessible. Big thanks to the PMTC team, our Spotless members, industrial members, as well as the people who have worked on the guide, and thanks for the funders. Thank you. Take questions if there's no questions. What? what's going on. Trying to open. Yeah, it's open.
Okay, so maybe just introduce Nicholas Aramuni is going to speak more on the cleaning research theme. Nicholas works with RABA in this area.
Thank you, Sarah. Thank you, Rabba. Good morning, everyone. Thank you very much for being here. It's a great opportunity to talk a little bit about what we do in the cleaning theme. And I'll just take about 10 minutes to give you an overview of how we did the experimental work as part of the Spotless project. So talking about optimizing the cleaning procedures at bench scale, and then moving on and translating to the pilot plant. So as Rabah previously explained, our process looks at first studying the residues themselves. So what chemical entities, APIs, formulations, etc., are we trying to clean? What are their characteristics? What's their solubility? So we do a lot of characterization and some computational work as well on that, on determining the best solvent mixtures, best detergents. Then, once we have all that set, that guides our laboratory cleaning tests. So we use the developed film flow cell, so the bench scale cleaning rig, we call it, where we test different process parameters and different cleaning agents that we have pre-selected using the computational study. And in this approach, we're complemented by the use of PAT, which inform us on the mechanisms that govern cleaning, and CFD, which also guides our flow rates, our selection of flow rates, and gives us a lot of information on the transport processes that happen. So what happens in solvent, how soil is being conveyed away from the surfaces, what's the forces applied on the surfaces, etc. And then once we have all that set, we've selected our best process parameters, our best cleaning agents, be it a solvent mixture, or a detergent. And then we translate that to the pilot plant, where we validate the scalability of the developed chemistry. And we test, or we check for engineering challenges that occur upon scale-up. I'm talking about, for example, deposition of sediments in the pipes, foaming in the tanks, the effect of different pumps, crossing in the valves, etc. There's quite a lot of challenges that might happen upon scale-up, so that's a very useful tool for us to validate that what we're developing or what we're proposing to industry can actually be easily replicated on a larger scale. So speaking a little bit about the bench scale, so we started off with the first picture on the left, which is a dipping test. So that's a very gentle cleaning testing technique where we screen purely for the chemical action. So just temperature and solubility or dispersibility. So we have steel or material samples, we call coupons, that we coat with an API or a chemical entity, and we dip gently in different cleaning agents at a set temperature. And that allows us to compare the chemical action of each. So that's a very fast screening technique, but that's just an informative first step, but it's not really replicating what happens inside a process vessel, let's say under CIP conditions. So then we move next to the film flow, where this film flow cell was actually designed to replicate the film flow that happens within CIP. So basically, it's a falling film that develops on a vessel wall, and that's the weakest part, or the weakest mechanical phenomenon happening in a CIP vessel, because you have the jet coming from the spray ball, and then you have the film flow falling on the walls, and that's the weakest part, so that's a bottleneck for cleaning. So if we investigate this, this allows us to optimize the whole process. And we use in situ PAT and visual analysis to also help us understand different mechanisms governing cleaning. CFD also complements this work. It gives us information on the transport mechanisms and guides us a bit on how to select the best flow rates, what flow profiles we get. And I'm showing as an example here, that's a snapshot from a CFD simulation by Aris and Mohsin, which shows you what happens to your shirt when you try to wash a spoon in your sink, a big splash. This is a jet impinging on a vessel wall. And then once we've done all of that, we've selected the best process parameters, the best agent. we scale up to our pilot plant. Now, under the film flow, so what we do is we use UV-Vis inline to measure the concentration of the dissolved species in the solvent. So that gives us, by integrating that, that tells us how much of the soil that's on the surface has been dissolved with time. And by looking at taking an image of the coupon under cleaning, so just like the videos that Traba has shown in his slides, where we see the cleaning happening, by analyzing the images, we can actually measure the surface area of the stain and see how that changes with time. And we can more or less relate it to the weight. So that tells us the total amount that's been removed from the surface. And by comparing both, or by subtracting them, we know what's the relative importance of the solution and all the mechanical phenomena. So mechanical removal, we can call it shearing or lift-off, but basically it's everything that's not the solution action. And we can study the relative effect of both phenomena in function of our process parameters for different cleaning solutions. And one of the interesting finds that we had from this study was that in cleaning processes where the dominant phenomenon is mechanical removal more than the solution, sometimes it can be very efficient, but it's always plagued with very high variability. So the reproducibility of the results are less. So although it's sometimes a compromise to rely on a more solubility-driven cleaning process, in terms of validation, it's probably more advantageous. So as a case study, we looked at developing heat maps for cleaning an API with detergents. So we screened for detergents, and we selected one detergent for the study, and we studied the effect of flow, temperature, and detergent concentration on the cleaning time and the cleaning percentage. So by measuring the weight that was removed. And we also did swab analysis on the surface after cleaning to validate that it's actually clean. And we were able to identify the sweet spot that Traba highlighted and prove that actually under optimal process parameters, it falls below our cleaning limit. Then finally, we scale up our work and move to the award-winning 8x rated CIP rig. Sorry, I have to take this chance to brag a little bit. So we haven't had a chance to actually describe it, what it looks like. So we have in that plant 2 holding vessels that are jacketed, where we can hold either detergents, pure water, or solvents. They're vented, and the whole system is 8x rated. So we feed those to our CIP testing vessel, which is the small vessel on the right you see in the picture. So that vessel, in that vessel, we can investigate different materials of construction, because that vessel is actually built of 3 parts that are interchangeable. So we can swap out middle sections, so we can coat them with specialized coatings, Teflon, steel. We can look at different materials of construction. We can look at different APIs, and we have different flow configurations as well. So we can either operate on the recycle or straight through. And we can modify the spray ball geometry, so the position of the spray ball in the tank, the type of the spray ball. So it's a very, very versatile rig that we've designed specifically to be able to replicate as high a number as possible of scenarios that might happen in pharmaceutical plants. And this rig is equipped with PAT inline, so we have a UV-Vis inline downstream of the vessel. So that allows us to look at dissolved API when we're doing solvent or aqueous cleaning in real time as well. So the same information we get on the bench scale, we're able to get from this pilot plant. But also this tells us a lot more about the suitability, let's say, of different pumps for when we're pumping detergent solutions, if there's foaming, pressure drops. So that's a very useful tool for us to validate the scalability of our methods, because we might, for example, select a flow rate that is maybe too low or too high on the bench scale, but would that translate to the plant? Would we be able to form a film efficiently at the flow rate we've chosen? Would the pressure drop in the lines be sufficient? So I'll wrap up quickly. So what we're doing here is the first step is design qualification. We can also do that. We do riboflavin coverage tests, and that allows us, so we wash off riboflavin from the vessel, and we look at it with a UV torch, and that tells us if there are any hard to clean areas, and we've identified a few, and we're actually in the process of changing some parameters in the rig to address that. Finally, I don't have time to show you this video, but we did a test cleaning paracetamol, for example, and that's the PAT, the UV profile we get from the vessel. So we have an initial cleaning spike, then a steady state dissolution removal, and then a final decay once the tank is clean. And if we compare that to what we get in a film flow cell, we see that for easy soil, which is lightly coated and easily soluble, we have replicability, which is good news as a start. So in conclusion, the Spotless project basically looks at a comprehensive approach to optimize the cleaning process, starting from computational work, solvent screening, and then development of the optimization of the process conditions on the lab scale, and then translation to the pilot plant. And basically, we predict that it will be very impactful for the pharma industry. So I'd like to thank first the experimental cleaning team and the Spotless industrial members. And finally, this is the overall cleaning research team that's been involved in this project. So with that, thank you for your time, and I presume we can take any questions. Thank you.
Just take a moment to introduce Dr. Oris Chard, who's working in computational fluid dynamics, lecturer in transportation and fluid dynamics system. You speak to it better than I. Yeah, so over to you, Oris. Thank you.
Thank you very much, Sarah, and it's a pleasure to have a chance to speak in this room. I know after a few years, the only opportunities I usually get to speak in this particular lecture room is when I'm teaching second-year students about product design. So it's a very different audience today, and a pleasure to have a chance to introduce and present some of the work that I've been doing in computational fluid dynamics, both within the Spotless project, which recently wrapped up, and now within the Impress project. What I'd like to talk about today, give you some of the background, some of the theoretical background about how we predict cleaning rates within vessels and within model systems, such as that film flow rig that was presented. So give you some theory background on top, chalkboard, not chalkboard talk here, but some of the theory. Then look at computations. So how do we use numerical schemes to solve the equations of fluid mechanics and make predictions about cleaning rates? We'll introduce then the model system, some details about how we scaled down from full vessel scale to a 2-inch wide flow channel. Give you some sample predictions, sample results for the computed fluid flow, and predictions of the cleaning rate, and then a few slides about what's called the Impress project, so simulations of sprays in vessels. I'm sure many of you who work in cleaning are familiar with Sinner's Circle. We look there at the different phenomena that contribute to determining how cleaning progresses. Does a particular process involve high temperature to improve the rate of cleaning? How long does a given process take? Does it rely on mechanical forces, shear stresses that are applied to a surface? Or does it perhaps more rely on chemistry, so dissolution or reaction of the soil? What I'd like to do, and the general goal of the computational fluid dynamics theme within the cleaning part of PMTC, is not to have qualitative judgments of the relative importance of these different phenomena, but rather to quantify them. So make predictions about what is the rate of cleaning due to mechanical effect, what is the rate of cleaning due to chemistry, so due to solubility, and what we call transport processes. What have we done over the last few years? CFD within PMTC started with the EI IPP project, 2017 to 2019. Some of the highlight images there where we studied a jet impinging on a surface, flow within a T-junction to understand rates of cleaning in the dead end of this junction, cleaning rates within a stirred vessel, and a film on the upper surface of a vessel. So how does that fall down the upper surface and then detach? Since then, we've had the Spotless project. You've seen the experimental side of it. What I'd like to do today is introduce the simulations. How do we predict the fluid flow coming down the coupon, and what is the cleaning rate? And now we've moved on to larger scale. So we started with exploratory studies of full industrial scale problems, looked at a model system, a 2-inch wide ramp, and now we're looking back at the full vessel scale. These are 4,000 or so liter vessels, and modeling the distribution of a spray within that size of system. Just to give you a brief reminder, what is happening when we try to clean a surface, and how does this solution work? If you think about putting a sugar cube into your coffee or your tea, perhaps this morning, I'd like to think about what happens the moment you put that sugar into your drink. If you don't stir, see that sugar cube falls to the bottom, the first layer of sugar molecules dissolves into the liquid, and then those molecules are stuck there at the surface. They form a saturated solution adjacent to the solid surface. And the only way those molecules can move away and allow new molecules to dissolve into the liquid is by diffusion, so by their own Brownian random motion. How can we accelerate that? The answer is by flow. We bring fresh solvent towards the surface, that displaces that layer of saturated solution near the surface, brings fresh solvent, allows faster dissolution now of the solid that's there, and we can clean the surface. We can dissolve the sugar faster. In terms of cleaning, that means we clean the surface faster because we have a fluid flow. So we can't rely only on solubility of the substance. Yes, that's the first step, and having a high solubility is very important. But in addition to having a high solubility, we need to have good transport of the solute that is dissolved away from the surface, and by bringing in fresh solvent. Briefly, what is computational fluid dynamics? Computational fluid dynamics means that we divide whatever problem we're thinking about into millions, maybe billions of small cells. So if you think about this room, we could grid it down to perhaps tens of centimeters, a 10-centimeter grid, maybe centimeter-sized grid, depending on the problem we need to solve. We break up the full simulation, the full system that we're solving into very small cells, and in each cell we solve the equations of motion. So think Newton's laws. Force equals mass times acceleration. How does the fluid accelerate? Because of the forces acting on every cell due to the adjacent fluid. We work with mixtures, so there's gases involved, there's solid surfaces and liquids. What we need to do is keep track of what is occupying any given cell. If we think about liquid and gas mixtures, we have to track that surface and include surface tension. So we know surfaces have a tendency to contract because of surface tension. We have to include that effect in these simulations. So as an example, you saw that impinging jet. I'll try to speed it up a little to see the interesting part where it hits a surface. So sample calculation where you do take into account surface tension formation of those droplets as they hit a surface. What do we use CFD for within cleaning? We want to predict the fluid flow structures and the cleaning rate. So how do we bring fresh solvent past a surface? What's the resulting cleaning rate? The model system we work with paracetamol largely because it's well characterized, relatively innocuous, only a painkiller effectively for us. So good to work with in the lab and good to work with computationally because we know the solubility that we need to know and we know properties like the diffusion coefficient. In terms of scale down, we can take a typical flow rate for typical pressure through a spray ball, take that total flow rate into a vessel, divide it by the circumference, and we get how much is flowing down a 2-inch width along the side of a vessel. So if we have 1 liter per minute, as you saw approximately from Raba's and Nicholas's presentations, 1 liter per minute over 51 millimeter wide channel is 123 liters per minute for a 2 meter diameter, roughly 4,000 liter vessel. Sample results showing the effect of wetability. If we only inject about half a liter per minute on this coupon and the contact angle is 68 degrees, representative of the polymer 3D printing that was used to make this slide, we see it doesn't wet that surface very well. In fact, it's not very good for cleaning. You need a higher flow rate to ensure that the whole surface is wetted. You're not just cleaning rivulets coming down along the circumference of your vessel. If we raise the flow rate, then we get full cleaning. Also, in this case, we don't care so much about the wetting because we know the flow rate is high enough that it fully wets the surface, so we drop the contact angle down to 0. We're not interested in the wetting effect, but if the flow rate is higher, we get full wetting, and then we get appropriate conditions to study cleaning. Comparing now some simulations at half liter per minute with the experiments, we do see that we get that double arch here structure. If you look carefully here, there's a reflective part, a dark part, and a less, a grayer part along the coupon. We see that we replicate that behavior in the simulations. What we're actually interested in is that dissolution effect that I've been talking about. So what we'd like to do is zoom in on what's happening if the coupon is fully covered with liquid, zoom in on what's happening right at the surface. So as I talked about the sugar cube dissolving, the same, that paracetamol soil on a surface or another substance dissolves into the adjacent liquid and gets carried down the flow. In the simulations, we see all the details of what's happening. We don't have to try to have a special microscope to see these things. It comes as a direct consequence of our calculation. So we can track, for example, how quickly does this bottom coupon get wetted. Here's the wetted area of paracetamol on the left. So how much we've covered over time. Of course, there's an initial delay before it rises up. The flow has to come down the ramp, reach the coupon, and then wet the coupon. In terms of the cleaning rate, we can characterize it in two different ways. The first question is, how quickly are we bringing molecules from the surface into the solute? Sorry, it brings into the solvent. And that's the red curve here. So we're bringing, first thing, the liquid reaches that soluble section, it starts to dissolve. We're bringing now a material off the surface into the flowing fluid. Once it has a chance to flow down that whole coupon, that's the blue curve. On the blue curve, we track how much of the solute are we detecting at the outflow of the ramp. And so eventually, of course, given that time delay, we have to establish the dissolution process and the convection, bring the liquid downstream. If you reach steady state, the two have to balance. You can only clean off of this surface as fast as you remove it from the outlet. And comparing, so predictions here quantitatively, we see we're predicting around a cleaning rate of 1.6 milligrams per second from that region, whereas in experiments we see around 1. So fairly good given the approximations involved here. Then I'd just briefly like to introduce what we're doing next. We've had very nice success in predicting what's happening along a small coupon, but what happens in a full-sized vessel? That's our next project, and that's the Impress project. Now scaling up, so now to even beyond, so near the maximum flow rate you could get through a spray ball, 1,300 liters per minute. We run these simulations with 2 spray balls now within a full-sized vessel. I show here a few snapshots. We only started this project a few months ago, just setting up the meshing and the geometry here. So we only have 0.8 milliseconds of a calculation at this stage. But still, we see already in this small time the establishment of the spray coming out of the spray ball and the development of the spray from that position. Would like to show you 2 videos. We have here those droplets colored by, this is actually up to 2 milliseconds now, those droplets colored by their speed, and, colored just showing their positions in 3D space, so it's a 3D volume rendering showing everything around. But on the right, we have a 2D cross-section, and the color here is the speed of the liquid. So we can see how it moves away at high speed, then as it moves into the surrounding gas, it slows down, it's dragged by the surrounding gases. So overall then, just to include here, we've been using computational fluid dynamics to predict the flow structures and the cleaning rates that occur within cleaning operations in pharmaceutical vessels. I showed you the effect of solubility, but it's not only solubility. We also have to think about flows. Does the flow wet the surface fully? What is the structure of the flow? How well can matter diffuse? How well can it be carried away by the solvent? And through that, we can learn how to clean better. Could we use a lower volume? If we flow faster, we might get faster cleaning. If we switch to a greener solvent, we might get slower, maybe diffusion and dissolution. But we can compensate for that by having a better cleaning structure, a better flow structure. Overall, we're working then to predict quantitatively the cleaning rates in complex geometries such as a full-scale vessel, and that's the main ambition of the Impress project. So with that, I'd like to thank my colleagues, Evgeny Berlutsky, a previous postdoctoral researcher who worked with me, Mohsen Mohimi, who is with us here, and he worked on most of the ramp simulations and the vessel simulations, and thank my colleagues, my experimental colleagues, Marina, Nicholas, and Rabba. So with that, thank you. I'd be happy to take questions, I presume, in this social meeting time in between the different sessions we have today. So with that, thank you, and I look forward to talking with you.
That's great. Thank you, Orest. And next I'd like to welcome Dr. Mike McAuliffe, who is one of our collaborators from CAPA, based down at MTU in Cork, to talk about a new core project which is just kicking off. Thanks, Mike.
Thanks, Sarah. So yeah, thanks very much, and thanks for the invite and having me here. I suppose Sarah mentioned already that this is part of the cleaning team. So it's one of the core funded projects that we have within the PMTC. So the project has a nice jazzy acronym called ARVIS. So it stands for Automated Remote Visual Inspection System. And I'll go through, I suppose, the initial kind of design and how we've gonna go about doing it. I suppose we're only a couple of months in as well, so just bear that in mind that there'll be some initial results when we do get to that point. So we have a couple of industry partners as well that are, I suppose, having input in terms of the asks and the design around the system. So they're listed there as well. So just to give a very brief overview on the project, so again, we've kind of touched on visual inspection, and Ravi gave a good overview of what it is and how it applies to industry. And I suppose the application that we're focusing on here is the support for cleaning verification in industry. And again, it's mandated that this is done to any vessel or production that has a product going through. So the ask by industry was to essentially demonstrate that an automated or a visual inspection system can confirm what is currently being done or can be seen by an operator or a trained operator. And again, I suppose we're looking for comparable levels of detection by trying to demonstrate the camera system to an operator. So the aim is basically to mimic the current embedded and accepted practices that would be, I suppose, established within various industries or companies, and again to, I suppose, be comparable to the human eye would be one of the strongest asks that we've been given. So just briefly touching on some of the short and potentially long-term impacts that an automated inspection system could achieve. So reduced cost, this would be, I suppose, heavily underpinned by many of the following topics. Cleaning deviation, so again, if you have something that went out of spec, you would have support based on previous data, digital captured images that you can go back then and look to support cleaning deviation. You can speed up that process as well. You'd have improved automated batch release. So again, you're moving towards, I suppose, digitization of your cleaning process so that you could potentially have this system built into your tank. Once a batch or a process is finished, it's held until the cleaning is done and achieved. And again, you can just improve that automated procedure, let's say. Maintenance and intervention, again, if you're recording and documenting your vessel line, you can go back to previous historical images and compare to current and look for any potential issues on the surfaces. And again, that's the support maintenance intervention. Reduced resources, again, it takes manpower to check a vessel if it's clean. You need to go through, you need to open it up, and again, that will have to happen for this system. But again, imagine that you can have this in a tank vessel where it can be similar to a spray ball, sent in, capture the inside of the tank. Again, you're saving time by not having to have an operator do that. And again, that's leading into the reduced time. And again, that's also linked with, I suppose, the overall process. You don't have as much downtime for the cleaning verification step. So, and again, that leads to some happy spaces and lots of happy faces. So, just to give some initial results and to kind of show how the approach that we're taking with comparing to a current system, and when I say current system, I mean the operators. So again, we've done a test where we've spiked or prepared samples with various substrate concentrations. We had 2 different observation positions. And again, this was, I suppose, guided by an SOP that had been provided to us by an industry member. We then assessed the stains and scored yes or no for pass or fail. And again, the 1 indicates a yes and the 0 is no. So you can see that the visual residual limit, the VRL, is across the board is approximately is 1 along here. So 0.2 micrograms per centimeter squared. Is what I suppose the operators have come back with. And again, that's tends to be the accepted standard. For the camera system, again, using the same substrates and the same spiked volumes and concentrations. We had a couple of different configurations on the system just to test it and to, I suppose, control the image quality, standardize the image. And again, you can see the results here that typically for the various lighting conditions, so the on-axis is basically the camera and the light are in the same direction. And we have a polarizer, which is the POL here. And again, that's just to help with reflection. And then we have an off-axis illumination. So again, that's when the camera is looking at the wall, but you're using lighting from the side just to illuminate at a different angle. And again, there is reasonings for that. Overall, the results are comparable. So you can see that the camera system has a visual inspection limit of 0.2 micrograms per centimeter squared. But we could achieve slightly higher if we used different lighting. And again, we won't include this in the system, and there's reasons behind that. And one of them is because we do want to test this on a live vessel. And again, the system we have went ahead with and our testing is limited in the sense that it is not quite ATEX rated, but it is up to that level that it is suitable for immersion into a vessel. Whereas if we were to start adding on light sources and additional controls around it, there would be limitations as to how much we can test that. So again, only those in the front row maybe might have an advantage here. So these are images captured by the camera. And again, there is a poster, I'm not sure where they are, but there is a poster that has better images of these results here. But again, you can kind of see there's staining forming here, and you can kind of capture that with the camera. And again, these are really scaled down images. But again, you can see that we have on-axis, off-axis, and again, you can see the effect of the lighting has on the substrates. You do get to a limit where you can't see anything, and again, I don't expect it to be visible here, but by inspecting the images after, it is possible to actually see the stain still. So again, that's the progress we've made so far. And I think that's it. I promised Sarah I'd only be 8 minutes, so hopefully I have. Thank you.
Thanks, Mike. Obviously, those looks I was giving you were working. I'm delighted now to introduce our panel, and it'll be chaired by one of our industry members, Kenneth Pearce from Hyde Engineering and Consulting. I'd like to welcome back our own Rabba Morris and some of our industry members, including Kevin Lydon, Eli Lilly, Tony Gray, from MSD Ballydyne, and all the way from the US. I'd like to thank very specifically Paul Apolito Steris for making the journey. So with that, I might just, as everybody's getting settled, I mentioned at the top of the session that we have some polling questions. So to everybody in the audience, I'd like to get a sense of who we have here today and the sense of the understanding. Do you as an audience have a good understanding or experience in cleaning validation? So hopefully you can see the polling question. Eventually I'll see the results of the combined physical and virtual audience here, I believe. And that's just useful information, I think, for the panel to have and to understand you know, who's part of the audience. Great. So we have a lot of knowledgeable folks. Okay. I'll hand over to you now, Ken.
Thank you very much, Sarah. Results are coming in. All right, well, while those results are coming in, I just want to say that it's great to be here today. It's great to be back in person with a room full of people all excited to talk about cleaning. That doesn't happen very often. We're a little bit weird in this room, but that's okay. We embrace the weird. So we're here, our topic for this panel anyway is challenges with upscaling of cleaning chemistry, and this is one that particularly close to my heart. We have our own development lab as well. We work on similar projects to PMTC. It's great to see it existing in Ireland. Normally we're trying to outsource this to the US, and it's great to see that it's existing here now in Ireland, and we're really excited to be involved with PMTC on that. But it is a constant question that we all get asked as industry, as academics, as partners, consultants. What does your testing at bench scale really mean for us when we step into a vessel? So we're going to go through a couple of different topics here, but what we really want is, okay, we do have, yeah, we're about 50%. I'm going to call that 50% of people here in the audience are involved directly in cleaning and cleaning validation. So there is a lot of knowledge in the room, and we'd really like to get questions from you guys as well, get a steer on where we're going, because there is a lot of knowledge up here, and we want to find out as well what the challenges are. You know, the PMTC are interested as well in what you're experiencing, because that's going to guide the things that we're going to work on, or that they're going to work on next. So if we can all kind of talk about it openly, we'll make more progress together. So I'm going to start actually with Tony, and one of the first things we obviously have to deal with is that cleaning chemistries now are not the cleaning chemistries that we were dealing with 15, 20 years ago. Even in the last 5 years, we can see an acceleration in the change of type of cleaning agents that we're using. Even solvent-based industries are looking at, is that the right thing to do? Can they change? So just I'd like to get some reflection from you on that side of it, and what challenges that poses, again, for scale-up when we're looking at different cleaning chemistries.
Yeah, so I guess as you said, Ken, the chemistries are changing. So flow, I can see a lot of people are moving towards flow chemistry, become more efficiency in throughput of and greener chemistry. Also, biocatalysis is becoming a very common process as well, and trying to cut down your number of steps from maybe 14 down to 6 steps to bring a molecule through to API. And that offers a lot of challenges with respect to using solvents for cleaning. Majority of API companies, because of the complexity of the chemistry, using a detergent during cleaning could impact on the overall stability. You're creating new molecules that you don't know what's forming, and you need to understand the toxicity of the newer chemicals you're forming. So you need to figure out how you're going to challenge your cleaning using mixture solvents followed by detergents to address that. Also, with biocatalysis, a lot of the molecules may not have a chiral center, or sorry, may not fluoresce, and you can't use UV. So you have to use alternative methods to detect that compound. It can be challenging. As well as that, the whole, and as I said, toxicity with potent chemistry as well. A lot of companies are moving towards more potent. That's the general way products are going these days. The whole containment and whether your methods are practical, verifiable, and achievable, and to meet the need of cleaning, and whether your plant can be a multi-product plant versus a plant that has to be dedicated.
Yeah, and I think as well it's important to be aware that with the changes in the manufacturing techniques, that obviously means the equipment is changing as well. So it's, you know, even in the API industry, it's not throw 200 liters of methanol in and boil it and recondense it. Yeah. And, you know, we're moving way beyond equipment that is even operating in that space anymore. Correct, so that obviously puts pressure on our colleagues in the cleaning chemistry industry as well to find agents that work better in those spaces as well.
Even with your equipment, if equipment changes, your spray balls change as well. So you're trying to reach at areas that traditionally before you wouldn't worry about, and now you have to worry about. And then you're looking at more the design, come back to design phase, and working with engineers to help design your equipment better, as well as other colleagues then around better detergents, better solvents to do that work for you, compatibility and so on.
Absolutely, and that's something we're seeing as well, is that it's back to us as the engineers to make sure that clean by design is now, it's a phrase we're all starting out with before we even think of a new facility or a new process, is ensuring that we've got our clean by design parameters correct from the very beginning. Moving on then, Kevin, I think complementary to that is then obviously the work that we're doing on automation of our processes, and I suppose particularly relevant to cleaning. And we've had an introduction to Arvis now as well, efforts in that area from the PMTC. there is obviously with automation, there are clear benefits, possibly from HSE perspective, but there's obviously challenges as well from the PAT perspective. If you want to automate it, you've got to have the correct monitoring systems and the validation of those and the setup of those and the confirmation from bench scale upwards is obviously a challenge as well.
Yeah, it certainly is. Now, I would see it that we still are all grappling with, what is the holy grail? We haven't found it yet for cleaning. We still all have our struggles with cleaning. I suppose I would see automation as the holy grail with PAT. I mean, if you have that, you've more predictable cleaning, and because of the challenges Tony was talking about with the new processes, more potent, you don't want the health and safety concerns of opening up vessels, putting your head in, looking at visuals. So one of the reasons, we'd be involved with Michael and the Arvis project is obviously you want to maybe get a faster, more reliable visual inspection, but there's a health and safety angle to that as well. Can we avoid opening up vessels, putting people inside or having putting your head inside with your torch and mirror? Can you just put a small camera inside, have it permanently in there, or at least be able to put it in through a small port and then release your equipment that way from a visual inspection? So I do think automation seems to be the way to go, but it's the buy-in then to do that, especially if you're like myself, with you're on older equipment, older rigs, manual cleaning is the norm, and then getting that to the level where it needs to be to automate it is a challenge from both resources, time, people. So at least I think the work then we do with PMTC, can you prove the automation actually will give you something to maybe build your business case? And then obviously the cleaning by design you mentioned, Ken, that should be for any new equipment we're buying, I would hope, or any new building we're constructing.
Absolutely. And since we do have so many people familiar in the audience, one of the, I suppose, issues from the automation side is, as you said, getting buy-in from your sponsors, from the company, that yes, we're going to put the effort into cleaning validation, that we're going to fund your PAT. But inevitably, because there's been a reticence there to do that for the last 10 years, I can think of only a handful of companies who've gone whole hog and bought into it and invested in it from the beginning, or even retrofitted it. Is there anyone in the audience who has gone back and added PAT technologies such as UV or TOC or even online bioburden, which most people will quake at the thoughts of doing that in outside of the API industry, but it does exist and it has actually been done. So is there anyone in the audience who has gone through that with their company, and was there any particular challenge that you came across on the validation or implementation stage that you want to share with us or ask a question about if you're thinking of doing it? No. Yeah. Oh, there's a mic, sorry. Yes, great, thanks.
It's just, I suppose it's just more of we, I suppose, to some degree use inline TOC analyzers for assessment of pipework, assessment of CIP return lines, things like that. But how our concern would be the sensitivity of inline TOC analyzers. Do we feel that the technology is moving enough in terms of sensitivity for TOC levels? Our concerns would be the level of detection on our inline TOC analyzers or something like that.
That's a good question, and it's one that you do get challenged with, and that's exactly what we're talking about. It's the scale-up issues. So on a bench scale, we know we can get 50 ppb if we're really good at it, but generally kind of 100, 150 ppb. Paul, do you have any personal experiences on that? Because I have one that I can share I can divert to you first.
Add some thought, and I think it kind of comes with some of the earlier talks on really characterization of the product that you're manufacturing, and also understanding of the detergent or solvent that you're using cleaning. Anytime that you're incorporating a non-specific method like total organic carbon or TOC testing, you have to factor that in, that you can't rely that to one specific element. As you start to look at UV or FTIR or Raman, it can be much more specific to the cleaning agent or to the drug active that you're manufacturing. So you really have to characterize what you need to clean and then understand what the solvent is or the cleaning agent that you're using, and also what is the health-based exposure limit, and then the concerns with the method that you're applying to show that it's removed to a safe level. So I think as you look at that holistic approach, then use of a non-specific method like TOC can be really nice. A lot of times I'm seeing those, they're kind of at line, so they're pulling a grab sample and they're making a measurement, as opposed to something like UV that was talked about earlier, or something like conductivity, in which it's truly in line and you're getting real-time measurements for that. But yes, I've seen TOC being implemented, and the cost of that, the instrumentation is getting lower and lower, so it's more practical, even for something like a parts washer.
Yeah, and I suppose your points on understanding your cleaning agent and particularly your H-bells are very important here. So knowing those limits, but on a project that we did in the US about 2 years ago, where we did a full-scale implementation of inline TOC on the return of all of the CIP systems, one of the things that we were able to develop as part of the characterization, and it even kind of surprised us, I'll be honest, but there's a short case study on it which we're hoping to squeeze into the next version of the Good Cleaning Validation Practice Guideline. But one of the things that can be developed, and this is where the technology really starts to show its benefit, and it's only as these cases are coming out now that we can start to rely on it, is that from the cleaning cycles as they were being characterized, as they were going through that qualification process, before we even hit validation, we actually were able to, because of the speed and sensitivity of the equipment, we were able to develop essentially a fingerprint per system. So it wasn't even that we were achieving the correct limit at the end, it was that we could see in the rinse out profile that this system behaved like this, and the next media system behaved like this, and it had a little shoulder. And that was not only a one-time occurrence, that was a repeated occurrence. So I think that's one of the real benefits of getting those methods and scaling them up. Yes, maybe you do lose a little bit of sensitivity, but if you've got something which is a fingerprint and you can ID that to a system and you can build that record, then, you know, and you've done your proper investigation to H-bells and you understand that, you can really bring that together as to the genuine point of fully automated release. You know, that capability is there if we're willing to put the development into it and take that first step. So there are companies who are taking those steps, and the rewards are starting to really show. So I suppose that comes nicely onto one of the next things that I wanted to bring in, which is this fantastic new ATEX cleaning rig, which I think is fantastic that you got the award for it. It's well deserved. I know it's been a long struggle to get there, but obviously that's something that, if I remember correctly, you have incorporated the ability to develop these PAT technologies alongside it, so as part of those cleaning developments, correct?
Yeah, definitely. So CIP rig, that was actually the design of that and the input, and what we have actually can be attributed to our members, because before we spec that, we surveyed all our members, and our SAB, the Scientific Advisory Board, they have their input, because we wanted to have a system that would help our members solve the current issues and maybe the future issues within the cleaning. That's why it's modular. It has the PAT tools, but what we have there is we have selected the UV-Vis for the sensitivity as a specific method, which was great. We have the conductivity and pH, and the funding was not enough to get the TOC as well, but it's planned, and hopefully we can
help
get
from
that.
Industry accepted.
Yeah. So, but it's planned to be added on, and it gives us the chance to work with our members and to try all those technologies and see if they are, possible to use or not. But our first trials were really positive, and will be detecting below 0.1 ppm, and I believe this is far below for most of the API. Maybe not for the highly potent ones, and maybe we can have a discussion around the other challenges in the highly potent drugs or whatever. But most of the products that we cleaned are above the 0.1 ppm, the technology has moved, and the sensitivity is there for some of them, because we tried the Raman FTIR, we tried the NMR, for example, but still, if we are looking to the sensitivity, maybe they are not ideal, but sometimes maybe you can get away with that if you want to understand more on the cleaning rather than the verification, so they will be really helpful.
And I think you've touched on another good point there, which is it's coming towards the sustainability as well. You know, so how are we developing these small scale processes, and can we really use the small scale processes to develop large scale sustainable models for cleaning? And I think that's really changed in the last number of years. I know 2016, I couldn't convince a plant manager to save 1 million liters of PW a week. Just by little tweaks, a day or two extra here in your cycle development or your atch scale or your bench scale testing. But I think that's also a challenge that's probably coming back to you guys as well, Paul, in terms of cleaning agents and working with companies for those small scale developments and the sustainability of those processes, and looking at lower temperatures, more efficient cleaning. Is what direction is that heading in for you guys at the moment?
Well, I mean, that's a great question, Ken. I think it's changing from site to site in terms of what the driver is in regards to sustainability. So something like decrease in temperature may be wonderful for reduction of energy, but it may create a microbial risk if that cleaning step is used to mitigate that concern. Going with the real-time measurements is wonderful because you can monitor when that cleaning process has stopped, and you're not relying on a fixed time. So therefore, you could reduce time and water as part of that cleaning, and which is really nice. And we've had another site that looked at phosphate restrictions and trying to mitigate a component. And when you're looking at mitigating something like phosphates, then you do not want to remove phosphates from the manufacturing process, because that's the drug product that you're manufacturing. So you don't have that opportunity. So you have to look at, are you using phosphates in waste neutralization? Are you using phosphates in cleaning? And then drive it out of those areas in which it doesn't impact the manufacturing of the product. And then looking at some newer alternative chemistries to clean that may not have phosphates but can perform as such. So I think that's a great question, and it varies from site to site on what the driver is to improve sustainability regards to cleaning.
Yeah, absolutely. And I think an example I've had of that recently, and I think we worked with your colleagues here in Europe and trying to find a solution to one of these. But like that, we put the work in at small scale. We found 2 cleaning agents. Boom, everything works. We've got this nailed on. And then we went back to the local council to get approval for the wastewater discharge, and there was a chelating agent in it. And as soon as we took that chelating agent out of the cleaning agent, boom. So the sustainability challenges and the small scale development work is really critical for things like that, finding out if okay, it works. Why is it working? And is someone going to stop me using that I'm not aware of down the line? And I guess, that's, all of those are particularly a challenge, Kevin, with legacy sites. So trying to make any improvement on things like this with a legacy site where you're possibly restricted by your utilities, you're restricted by a site or a facility that is already full of pipework and full of everything already, and bringing on new systems can be a big challenge there. So I suppose accurate small-scale development makes a real impact.
It does, and I suppose, Ken, are you asking about legacy products and then how to make them greener? Is the cleaning greener? Is it?
Yeah.
So yeah, that's a huge challenge, I would say, because you have a robust cleaning validated process, so changing that is always going to be a huge effort. So if you do want to make it greener, I think, you know, you might be starting off with, well, I don't want to maybe change to a completely green solvent yet, but, you know, if I had some PAT online, can I do that? And, you know, can I reduce volume, you know, that I'm using, if it's solvent or detergent, and kind of come at it, and I think in a stepwise way. And then you might get to a stage then where you have your data then for the new green solvent, it's stable, I understand it, there's no impurities that are formed as part of it. And I think doing that, you might be able to change over. But it is a big ask, but I think there is room today for us to, you know, understand our processes more by putting more PAT on and then reducing volumes, you know, because I do think we kind of automatically default to throw everything at it so I get my cleaning right first time. And that's been fine for a number of years, but I do think there's a step change there we can all take.
Absolutely.
You're limited though with your site, you have a legacy site, you can't, not been given the funding. Using the work that Rabbit did around the design of experiments, trying to find that sweet spot of like your center circle, you know, your temperature, your volumes, your time and so on. You can tweak your cleaning as best you can and improve your stepways. We're getting there, but you know, you convince people to go with you on that road as well.
Yeah.
And I think there's huge benefit to be gained as well of forums like this, where we're all in a safe space to talk about cleaning together, and we can share those experiences. So, you know, there's plenty of knowledge up here as well. So don't be afraid to talk to us during the day or ask us questions when you do see it, or share your experiences. We'd all love to hear it and learn from it as well. We were sharing discussions over coffee this morning as we were waking ourselves up and getting ourselves ready, and there's lots of interesting things happen. But I think also one of the challenges then on scale up, and particularly where we're working on automation, and I know, Tony, there's automation projects going on your side at the moment as well. But I think one of the additional strains then, where we're optimizing our cleaning and automating it to such an extent, that puts an additional kind of training and knowledge impact as well on the site that we have to deal with in terms of operators, them understanding it, and them also, it's a totally different set of issues that they have to be aware of and be watching out for that they're maybe not used to historically.
Yes.
Oh,
You got what you wished for. We have a question, I think, from somebody online.
Brilliant.
So Stephanie, maybe you could articulate that.
Hello, yeah. So Steve Jeffrey said, for reduced fluid use, seen some recent discussions on burst cleaning, rapid wetting, and then periodic impact, soak and rinse. Has the panel any experience of using burst cleaning for improved cleaning results and reduced fluid use?
Maybe I just
can
title those comments and then leave it to...
Yeah, I'll add a little bit. So something like tablet coatings is something that requires time to wet before you can kind of remove it off. So by using bursts allows you to minimize the water and allow wetting of that residue. One of the things that you want to be cautious about high pressure and burst cleaning is if you're having a recirculating system, is making certain that it doesn't come off as large enough particles to cause issues with the spray devices. So making certain that you're using like a pressure monitor on the before the spray device to pick up that, or utilizing a single pass to make certain that the larger particulates go to drain before you start doing recirculation. But bursting can be very effective to reduce volume and also to kind of improve cleaning as you're moving through different circuits. Ken, I don't know if you want to add anything to that.
yeah, just a small addition to that. Your point about understanding the flow mechanics is really important with burst cleaning. You know, if you're operating on in the wrong space, you can either not achieve this burst impact you need for your spray devices to function, you know, to actually achieve their coverage. But you can also create knock-on effects for other paths as well. So if you're switching between paths in burst, that can be problematic as well, if depending on how your system is constructed, whether it can absorb those impact changes, and whether you've got the appropriate system. But it's definitely a way to take learnings as well from the small scale and bring it up. So we've implemented in several circumstances where, like that with tablet coding, or other circumstances where there's a soakage time required for it to start to activate, or where there's a very long contact time required, but you've got maybe 4 sub-circuits within a cleaning cycle. So how is the best and most effective way to achieve those 2 criteria? Well, burst cleaning and toggling of flow paths, and actually building that into it from the beginning, not saying I'm going to open flow path 1 and run that for 15 minutes, but working through them, you can actually achieve an even greater time of contact over a shorter period, build in your soakage, and it can all be good. So there's definitely benefits to be gained from that in terms of delivery, but characterization at small scale will give you the information you need to really get that, as well as making sure your engineering team are on the ball. But I think we're running
up on
time. We're
because
way
I
over,
know
probably.
We've got coffee break. But what I would like to do is would like to thank everybody on the panel and all of our presenters. I think really insightful discussion. And before I release everybody to the coffee break, I think maybe just one more of the polling questions. I think it's useful to understand from what you've heard today through the presentations and the panel discussion, if you had access to this pilot scale facility, how would you most see the benefit of that in your organisation? So we might just take that question. And what I'd like to just highlight as well, we have a poster display. If you're exiting on the at the back, at the top of the lecture hall, you'll see the poster display. Coffee is served there, there's restrooms, and we do have coffee and breakfast served on the ground floor where you had entered. So yeah, great insights there from the audience. And yeah, we're back at 11:30. 11:35 at a max, but thank you for your attention, and we'll take the coffee break. We have many exhibitors as well, so do take an opportunity to check in there with our member companies. Thank you.
Thank you, Ken. Nice job.